Hello, welcome to this virtual presentation. My name is Elizabeth McCready. I'm an associate professor at McMaster University in Hamilton, Canada. Today, I'm going to be talking about a pilot study that we have ongoing to evaluate the clinical utility of the CytoSure Constitutional NGS assay for the detection of genetic variants that may influence phenotype in families with variable expressivity CNV. I'd like to start by thanking OGT for this opportunity to speak about the work that we have ongoing, but also to thank some of the people in our lab who have been instrumental and fabulous in their assistance with this project, particularly Patricia Wojciak, Landrina Fonsum, Barry Eng, Yoko Ito, and Nassim Vasily. Also important, thank you to the entire OGT team for all of their support. They've been fabulous as we've been trying to work through this evaluation project. In undergrad studies, the question that ignited my passion for genetics was this very question. There are over 3 billion base pairs in the human genome. How then can a single base pair change contribute to multi-system diseases when most changes in the genome are benign? Of course, it isn't as simple as that. And it isn't only single base pair changes that can cause disease. In fact, we all carry single genomic variants and even genomic deletions or duplications or epigenetic aberrations. And yet we are able to get up every day, and hopefully most of us are able to enjoy a healthy day before we start it all over again tomorrow. So, what are the exceptions that can cause some to have a clinical phenotype and others not? With regards to developmental biology, there are a lot of interconnected moving parts, including both genetic and environmental factors. However, with regards to neurodevelopment, we have known for some time that there is an increased burden of copy number variants in patients with intellectual disability and developmental delay compared to normal control populations. This was nicely demonstrated by Ko et al. In this plot that shows a decreasing number of large copy number variants in control populations compared to patients with intellectual disability and developmental delay. However, the interpretation of large CNV is not always straightforward. If there are a large number of genes involved, particularly genes that have established dosage sensitivity, then interpretation is straightforward, but it isn't always that way. Case in point. This is a case that we examined several years ago. This child had a number of issues. As a neonate, it was noted that he had short stature, congenital heart defects, low set ears with macrocephaly, some digit anomalies, and cryptorchidism. When chromosome microarray testing was performed, a 1.7 megabase duplication was detected involving material from chromosome region 2q13. At the time, there was very little known about copy number changes of this chromosome region. There are 9 genes duplicated by the CNV, but none of them are known to be triplet sensitive. There were also only rare reports of similar CNV in the literature, although, based on large published cohort sets, this CNV appears to be enriched in patient populations compared to controls. There are less than 20 cases in the literature of 2q13 copy number variants that I'm aware of. We had also reviewed the phenotypes in 6 additional deletion carriers and 10 duplication carriers. The reported phenotypes are variable, yet the most common phenotypes include developmental delay, nonspecific dysmorphic features, and neuropsychiatric conditions. While there are at least 3 de novo cases, most of the cases in the literature and our review have been inherited from a phenotypically normal parent, or at least an apparently normal parent, or from a parent with a milder presentation. So what does this mean for our case? Is the 2q13 duplication relevant in this child, or is it an incidental finding? Indeed, there's a growing body of literature that describes recurrent copy number variants with variable expressivity and reduced penetrance that are similar to the 2q13 CNV, but dissimilar to classical syndromic CNV. Notably, these CNV are often inherited from a normal or less severely affected parent. There's also often variable non-specific phenotypes that are reported in association with these variants. Albeit, the full range of associated phenotypes may be limited by ascertainment biases introduced through genotype to phenotype characterization of genomic variants. These biases can lead to underrepresentation in the literature of milder phenotypes that may be associated with a variant, but that may not come to medical attention. Moreover, with the classical syndromic CNV occurring almost exclusively in patient cohorts, the variable expressivity CNV tend to be enriched in the patient populations compared to normal controls. The 2Q13 CNV meets many of these criteria and is thus similar in many respects to other variable expressivity CNV, including the 15Q13.3 microdeletion, the 22Q11.2 microduplication, and those listed in the diagrams here by Jensen and Girjan. Jensen and Girjan nicely highlighted variability in these syndromes compared to classical syndromic CNV, and also highlighted an increased frequency of neuropsychiatric phenotypes in association with variable expressivity CNV compared to syndromic CNV. There are additional reports that have looked at these CNV, which confer an increased risk of autism, schizophrenia, and other neurodevelopmental neuropsychiatric phenotypes. This is particularly interesting one here by the group of Stansel et al. They examined 26 CNV alleles associated with increased risk for autism and or schizophrenia. They looked at cognition in schizophrenic patients. In control carriers with neuropsychiatric CNV and control carriers with other CNVs. They found that among the control carriers with neuropsychiatric CNV, they performed at an intermediary level between population controls and individuals with schizophrenia. This raises the possibility that, in fact, differences between patients and control carriers may be due to variable expressivity and not reduced penetrance, suggesting that apparently normal carriers may in fact still be part of a continuum of associated phenotypes, but perhaps at the milder end of the spectrum and maybe even having unrecognized symptoms. Similarly, Pizzo et al. Looked at a large number of families with variable expressivity CNV, and in particular at the carriers and the probands. They noted that there was more severe phenotype in many of the probands, but that many of the carriers also have some phenotypes. Generally, the phenotypes in these carriers were milder than the probands. They also noted that many of the carrier parents had a lower burden of additional variants across the genome compared to the probands, suggesting that additional hits in the probands may have contributed to their more severe phenotypes. The concept that a high burden of deleterious mutations across the genome can affect disease severity is not a new one. This is a report by Doherty and Owen. They also suggested that an increased number and severity of deleterious variants can result in more severe early brain insult and intellectual disability compared to individuals who may have fewer mutations and thus milder phenotypes. This has led to a two-hit hypothesis for neurodevelopmental disorders, whereby variable expressivity CNVs are often inherited from a parent with milder or less severe phenotypes. Reduced penetrance or genetic modifiers are basic concepts that have been around before and may influence phenotypes in affected carriers. However, the two-hit theory expands on this and suggests that the variable expressivity CNV in affected patients may result from additional hits, including deleterious single nucleotide variants, indels, or CNVs, which can have compound effects to act synergistically with the single genes or allogenic pathways and networks, resulting in a more severe clinical phenotype than if the CNV occurred as an isolated finding. So, what does that mean for when we are doing microarray testing? Should we be reflexing to look for second hits or not? This is clearly something that needs to be sorted out. I think that it is of increasing importance, particularly in this area of large gene panel, whole exome, and whole genome testing. What else should we be doing when we've identified these variants of variable expressivity? We need to provide better assistance in predictive testing and understanding what is happening in these families so that we can better cancel those identified with these types of changes. In our lab, we are interested whether identification of second hits can have clinical utility or not. We've been looking at how we may explore this. Fortunately, we don't currently have whole exome testing available in our lab. We also have a limited genome analyst and bioinformatics group. So we're looking for solutions to allow us to get to a point to explore the clinical utility of testing for second hits, particularly in cases like 2q13 and other similar variants. That is why we've decided to evaluate the OGT CytoSure Constitutional NGS assay as a solution to this particular question. The CytoSure assay is a comprehensive NGS-based assay that targets greater than 700 genes known to be associated with intellectual disability and developmental delay. This very large gene panel allows detection of single nucleotide variants, indels, and single exon-level copy number changes in these genes. But what was particularly attractive about this gene panel and why we wanted to evaluate it is that in addition to these 700 genes, there is a backbone of oligonucleotide bases that are spaced throughout the genome for detection of copy number variants greater than approximately 190 kb. This is of importance in a scenario like ours, where in fact we are also interested in copy number variants that may occur across the genome. We are relatively early in our evaluation of this gene panel. So far, we have tested 24 individuals. These are all cases that have copy number variants pre-examined in them. The copy number variants have been tested in all cases using a chromosome microarray, the Affymetrix Cytoscan HD array particularly. We tried to choose cases that had either a known deletion or duplication in the proband. When available, we also evaluated parents who were also known carriers. Showing on this karyogram are the CNV that we analyzed. We tested one case with a 1q21.1 duplication. There was also several cases with 2q13 duplications or deletions, several cases with 15q13 deletions, one with a 15q13 duplication, as well as 16p13.11 duplications, 16p11.2 deletions, some 22q11.2 deletions, including a central 22q11.2 deletion, and finally a 22q13.3 duplication. So several variants across recurrent copy number variant sites. This is an example of one of the cases that we had analyzed. It's a proband with a 16p13.11 duplication. This is the chromosomal microarray data from this case that we had obtained using the Affymetrix Cytoscan HD array. It shows the duplication in the center of the diagram with the duplicated segment overlapping the recurrent 16p13.11 duplication region. We also looked at this proband by FISH. This shows the duplication in a metaphase from the proband with an enhanced signal on one chromosome 16, suggesting that it is in fact a tandem duplication. We also did FISH in the mother, as well as quantitative PCR, to confirm that the mother carries the same duplication. This is the data using the CytoSure assay. You can clearly see that the region on chromosome 16 that has jumped up with an increased log2 ratio score. It is the same in the proband and in the mother. It actually very nicely jumps out here. So absolutely, this finding was concordant with both the chromosome microarray and FISH. And we're very excited to see this. This was, in fact, one of the first cases that we looked at with the CytoSure data. And we were clearly able to identify the CNV. And so we're very happy. That was good news for us and for the rest of the study. In fact, when we looked at all of our cases and the copy number variants that we were concerned about, there was a high level of concordancy. 15q13.2 to q13.3 deletion and duplication, 16p13.11, 16p11.2, the 22q11.2 variant, and one case each of 1q21.1 and 22q13.13 variants. They were all concordant with the chromosome microarray and seen absolutely beautifully with the CytoSure assay. They were fantastic. Looking at that data, we were a little bit concerned in the first analysis that several of the 2q13 copy number variants were not called by the software, but when we went in and looked at the raw data, they were absolutely all there. Why these 2q13 variants weren't called originally is being looked at, but clearly they're evident here from the log-2 ratio plots in each instance. Case C12 had a 2q13 deletion. There are 5 probes on the backbone that map to the 2q13 deletion region, and you could see that these dropping down here in these images. In case 21 and case 22, which is a child-parent duo, you can also clearly see that deletion. Case C19 and case C20 are also a child-pair duo with a duplication this time rather than the deletion. Again, it is the exact same probes that are popping upwards in the duplicated segment that were dropping downwards in those cases of deletion. This is all very good and consistent with our CMA findings, so we're very happy to see this when we looked into it. I thought that I'd take a brief moment to go over some of the other cases that we saw, specifically because one of the key things that we wanted to assess was not only could we detect the known CNV in our cases, but also is there anything else in these cases that can be picked up and may be relevant for the observed phenotypes. This particular case is an interesting one. It is case C4 that had a 15q13.2 to q13.3 deletion. And you can see that here on the OGT CytoSure data as a dip in the log-2 ratio score. This was absolutely concordant with the chromosome microarray data. When we started looking at what else we can see, this was an interesting hit here in case 4. This showed up as a copy number variant rather than when we were looking at the single nucleotide variants. In the image, we've zoomed into the Nerexin-1 gene. Note that there is several probes here that have a reduced log-2 ratio score that was called by the software as a deletion. This also correlated with the very low coverage of the exonic region, which would also be consistent with the deletion. We also took a look in this case at DNA from the mother. She does not show a dip in these specific probes, which suggests that if this is a real deletion, then it would be present in the proband, but not the mother. So this might in fact be a second hit in the proband that could be contributing to the more severe phenotype in the proband compared to the carrier parent. Unfortunately, this is a relatively new finding, so we haven't been able to confirm this yet. Our hope is to test for the Nerexin-1 deletion by a quantitative PCR method to confirm the finding, because it's a very small deletion below our level of resolution by CMA. So we're going to have to use a different method than CMA. For those of you that aren't familiar with Nerexin-1, it encodes a protein that mediates neurotransmission through formation of synaptic contacts. Nerexin-1 exonic deletions have been described in several patient cohorts and case reports in the literature. It is now well established that haploinsufficiency of the gene can be associated with variable findings that may include intellectual disability, speech delay, autism spectrum disorder, and seizures. This is a particularly interesting report from Lowther et al. that I'm showing here. They described 5' exonic Nerexin-1 deletions are likely more penetrant than the 3' deletions. In our case, the deletion is of exon 6, which is similar to some of the patients represented by hatched bars in the blue box. The cases with hatching in this report are cases with other secondary findings, which would also be similar to our case. Again, it begs the question whether the second hit in these cases is the additional genetic insult leading to a more severe phenotype in the patient, but not carriers. Another interesting case was case C12. This was one of our 2Q13 cases. There was a very interesting SHANK2 mutation detected, resulting in a nonsense variant that is predicted to disrupt protein function by either nonsense-mediated RNA decay or protein truncation. There have been a number of reports describing SHANK2 de novo exon deletions in patients with autism spectrum disorder, as well as de novo nonsense variants that have been described in the same population. There have also been de novo SHANK2 deletions that have been described in schizophrenia patient cohorts, often as a second hit. So again, this is another interesting finding that we will need to confirm by a secondary method, probably by single sequencing, but possibly by exome sequencing. Finally, I would like to draw your attention to one last case. This is a 22q11.21 deletion. You can clearly see this deletion in the OGT CytoSure data here. It is a deletion of the region between low copy repeats B and D, consistent with the central 22q11.2 deletion that includes CRKL. In this particular case, there was a 4 base pair deletion in the FLG gene that results in a frame shift of the coding sequence. In this diagram, you can see the deletion in the middle of the shown sequence reads, which is associated with a corresponding drop in the coverage indicated by an arrow. FLG haploinsufficiency is interesting in that it is associated with atopic dermatitis, allergy, and asthma with incomplete penetrance and variable expressivity. It is not necessarily a neurodevelopmental or neuropsychiatric gene, but we often do see features in these cases other than neurological specific ones. For this particular case, we don't have information about whether there is skin findings, but if this was a prospective case where the individual is being examined by a clinician, then that may be useful information to have. FLG haploinsufficiency can also cause ichthyosis vulgaris, which has a semi-dominant inheritance pattern. Again, that may also be an interesting piece of information if there's skin abnormalities in this case. In summary, there is an increasing evidence to support an influence of second hits on phenotypic expression, variable expressivity CNV. There's a growing body of literature on this topic. I think that we need to be more and more mindful about the possibility of second hits. As laboratories doing chromosome microarray testing or other genomic characterizations, we need to consider the complexity of our genome and recognize a need for tests that assess multi-layers of that complexity to better understand the extent of genomic changes that may influence phenotype. NGS applications can enable simultaneous detection of SNV, exonic deletions and duplications, and large copy number variants, and therefore may have utility for more complete characterization of the genetics in families with variable expressivity CNV. With that, I'd like to thank you for taking the time to listen to me today.